I think it's pretty clear that the kinds of things that vary, for the kinds of ingredients and the kinds of dynamics for which brains are sort of famous, that and this is why people think brains are associated with intelligence and so on. Those kinds of processes go on in every tissue of your body, right? So almost I'm not aware of any real distinction that one could draw between the kinds of mechanisms. People talk about magnetic fields, and they talk about quantum events in microtubules, and they talk about the electrical signaling and various hertz, different kinds of integrated spiking and all that. All that kind of stuff happens in other parts of the body. And so I think we have to be really open to this idea that treating the rest of the body as clockwork, whereas we are kind of okay with attributing various higher level cognitive properties to brains, I think that's a real mistake. And I think we're leaving a lot on the table in terms of biomedicine and other things by making that assumption.
Yeah. May I ask, how did you come up with the name scale-free cognition?
That possibly could have been, so that comes from a paper that I did with Chris Fields. I don't recall if he was the one that came up with that title or if I was. I don't remember at this point. He and I do so much together. I don't know. But the reason it's good here is that we as humans are very focused on a very much like we are in the electromagnetic spectrum and so on. We only see this like tiny little bandwidth, right? We are the same way for intelligence, and so we are very good at recognizing kind of medium-sized objects moving at medium speeds in three-dimensional space. And look, that's a crow, and this is an octopus, and that's an orangutan, and we can sort of recognize intelligence. But there are intelligences all around us at various scales that we just find very difficult to think of in the same way. So the bacteria in your gut that are solving physiological problems and navigating not just physical space, but physiological space in very clever ways, that's an intelligence, but it's very small. And literally it's very small, and it's hard for us to recognize those. The various organs that make up your body, people will immediately say, well, they don't have a cognitive self. I have a cognitive self, and they're just part of me. Well, yeah, that's because we and they would say the same thing about themselves if they could talk, right? So this is just basically a first person perspective from a cognitive system. And we're very bad at recognizing the different scales. Going in the opposite direction, one could, for example, and this is also something that Chris and others and I are working on. The paper's not out yet, but this idea that one could look at a whole evolutionary lineage, or in fact the process of evolution itself, as this giant distributed agent where every particular animal is a hypothesis that agent has made about the outside world. And like all of our ideas that are being sort of churned over by this active inference process, these hypotheses may be supported and they may survive and give rise to other such hypotheses, or they may be unfit and die and you'll never hear from them again. And so that's another, that's those kind of ideas. And I'm not saying it's right or wrong, but those kind of ideas, you can't even begin to have those kind of ideas and consider those hypotheses if you are tied into the fact that all cognitive systems are roughly the size and scale of us, and and they have to have the brains, and they have to work on a time scale that's too rapid, and that begins to look too weird and too stretched out, and well, that can't be a mind, but it's all relative. It's all completely relative. But we have this vantage point, like we used to when we thought we were the center of the universe and all that. We have this scale vantage point that just really obscures the reality of things.
Cool. Yeah, because I was wondering about the name, how you named it there, because it seemed like it struck me as more scale dependent cognition because it's reliant on the space and time scope. But perhaps scale free means, I don't know, it's like a different approach. It's like a macro look at it versus.
Yeah. I see what you mean, right? So you're right in that this approach does emphasize looking at scales. It doesn't. The point isn't to get rid of scale. What I think, when I say free, I mean it's kind of a play on words. There's two ways to think about this. One is it's free in the sense that it's not tied to any one scale, so you can sort of jump up and down, and it still works, right? It's these invariants. So the same thing is true and we see this all we see this all the time. I think physicists use it this way too. It's something that's true on one scale, and then you turn some control knob, whether it's size or time or something else, and exactly the same thing is true at this other scale, right? That same dynamic. So that's one. But
Gotcha.
I also like this idea. To me, what free really specifies there is freedom, in the sense of freedom for the system to do what it's going to do, and freedom for you as a scientist to not be locked in one particular lens of looking at the system. That's how I see things. I see things as, sort of unfettering our ability to recognize these diverse intelligences. Let me sneak up on it from a slightly different perspective, because I think stress is very important. One way to think about the behavior of these collective intelligences of cells, and we're talking about, let's think about regeneration or development, which is basically just regeneration from a single cell. Same thing. What you have there is a kind of what the system is doing is it's navigating in morphospace. It starts off in this, so morphospace is this a virtual high dimensional space of all possible anatomical configurations that a particular genome could possibly reach. And so you start off in one corner of this morphospace as a single egg, single cell, and then you sort of navigate to this other region, which is an ensemble of states that we recognize as the correct target morphology for whatever creature that you are. That navigation process has quite a bit of intelligence to it, in the sense that not just this is key. I'm going to take a quick detour here. But one way people critique these ideas is they, oh, well, all you're doing is painting the word intelligence on any complex process, right? Anything that happens, you say it's intelligent because look how complicated it is, and it got from here to there. So that isn't it at all. This is really critical. First of all, you cannot estimate intelligence from observational data. It has to be interventional experiments. And what you're looking at is not, you're not looking at emergent complexity. That's very easy. There are many systems that start off with very kind of simple local rules, and then something very complex comes out the other end. That's not intelligence. Intelligence is the ability to get where you're going, to reach that goal, despite novelty and perturbations, despite its problem solving, basically. So this is key. So you're this collective intelligence because you're a group of cells. Very early on, you become a group of cells, and you have to reach wherever it is that you're going. Okay, so the part of this that's intelligent is the part that when you deviate it from what it's trying to do, it finds other ways of doing it. So let's just use a simple example. We got a salamander, and in that salamander, there's a very particular limb that forms with a certain number of fingers. And if you amputate that limb, and you can amputate it anywhere from the shoulder to the fingertips, the cells will immediately grow. They will start to replicate. They will differentiate. They will do all these different things, and then they stop. This is the most amazing part: is that they stop. When do they stop? They stop when a correct salamander limb has formed. So the way one way to think about this, well, the traditional way to think about this is as feed-forward emergence. So yes, every cell has a bunch of local rules dictated by DNA and biochemistry, and you just sort of roll them forward, and whatever happens, and it just so happens that it's a salamander limb. So that's one way of looking at it. I think that way of looking at it is missing the boat in a profound way, because it limits our understanding and our ability in regenerative medicine. But anyway, the view that I recommend is that you look at this as a kind of homeostatic capability of this collective intelligence. What it has is, and by homeostasis, I mean the thing that happens in your thermostat, where there's this loop where it takes a measurement of something, let's say temperature in the case of your thermostat, it makes a decision relative to a memory, a set point of what the right temperature should be, and then it takes actions to get closer to that set point, right? So you can think of regulative development and regeneration and cancer suppression and metamorphosis in exactly the same way. There is an error minimization process that tries to work to keep the system in the correct anatomical region of space. And when it's deviated by injury, by mutations, by teratogens, by whatever, it tries to get back there as best as it can. And I give lots of talks about all the amazing examples of what it can actually do in order to achieve that, just all the weird new solutions that it comes up with to various problems. So in order to do that, so how do we think about, I mean, the key thing about that loop, that control loop, and this is, for engineers, this is absolutely nothing new, that control loop, the key is that it has a set point. And why does it keep, why do these cells act? Why do they spend metabolic energy? Remember, they used to be independent organisms. Individual cells
Mhm.
care about little tiny single cell goals. They care about metabolic state. They care about local concentrations of things. They don't care about these large goals of organ shape and organ size and things like that. So why do they keep trying to do this? And one way to think about this, and so this is an idea that I sort of push forward in that Tame paper, and then since then we've done a lot of experimental work on it, which is not published yet, which you'll be seeing later this year, and some good computational modeling of it, some of it which is published as of a couple of weeks ago. So one way to think about this is that what keeps the system trying to minimize error is stress. Now, stress is a really good word because stress has been used by engineers when they talk about various materials. So they talk about stress in mesoscale stress in materials. It's talked about by psychologists, it's talked about by evolutionary ecologists in terms of ecosystem stress, of course, behavioral scientists in terms of single, but where people don't really talk about stress very much is at this middle level, which we focus on, which is cells and tissues and organs. And I think that this concept of stress is quite general, scale free, if you will. And that the deal with stress is that one of the things, and there's a couple of major things, this is just one of those, one of the things that helps scale the cognitive light cone and move a set of subunits from caring about, from a pile of cells that care about individual cell level things to a collective that cares about much bigger things, like, hey, we don't have the right number of fingers currently, right? And no individual cell knows what a finger is or how many you're supposed to have, but the collective does. And the thing that keeps them motivated to kind of try to get back to where they're going is stress. And so what you need for this in that system, what you need for this is for every cell to be able to be stressed by and to spread its stress to other neighbors in response to conditions that are really failures of alignment at a larger scale. So, for example, so just to give you an example, if we were to take an eye and stick it on the tail of a tadpole, right, and sort of transplant it, you could have a situation where no individual cell is damaged, nobody's injured, nobody's poisoned, there's no DNA damage, there's no reason for any individual cell to be stressed, but the collective is at the wrong location in anatomical morphospace. There's a geometric problem. And so, or for example, after amputating the limb, after all the cells heal and there's, the skin, everything heals, no individual cell should be stressed because everything is fine on the single cell level. But there's a real mismatch there's a delta between the pattern memory that the system has, that's that set point, and where the collective is now. So I think, and there's a lot more than we have time to go into here, but that spreading of stress and that linking of individual cell stress response mechanisms, which used to be all about migration and various physiological responses, used to be about single cell stresses, things that could happen to an amoeba and stuff like that. And they're now getting harnessed to much bigger states of affairs. And that's the beautiful thing about these homeostatic loops, is that the cybernetic kind of aspect of it, it's a set of wildcards. Measure something. What do I want to measure? Well, it could be anything from the quality of your DNA to the length of the tissue in which you find yourself, to your position in the hierarchy of the tribe, some sort of social thing. Any of that can be the thing you measure. And then there's a set point to which you compare. And again, that could be almost anything. And then there are some actions you could take, which again could be almost anything. So what evolution does is it uses this basic loop, this basic concept of this loop, and then it just progressively harnesses the three sort of main parts of that loop and points them at different sizes, different scales, different types of things. And that I think is a major aspect of the kind of multi-scale competency architecture that makes this whole thing so robust and interesting.